Hello, and welcome back to part 3 of adaptive immunity. I'm sorry, I kind of had to cut off part 2 rather rapidly because my microphone was dying and beeping very loudly in my ear. So just sort of recapping what we were discussing. So we were comparing and contrasting the primary immune response and the secondary immune response in regards to humoral immunity. And so it doesn't really hurt. So looking at this graph, this is showing you when there is an antigen presentation, and this is when the immune system is naive, we call that a primary response. So notice in the primary response that there is a lag period. There's a lag period where you see nothing is happening. On this axis, we're looking at concentration of antibodies. So you see nothing right there. That's called the lag period, and that is because this antigen has been introduced. It needs to find its one true B lymphocyte, so that's the clonal selection. And then it has to go through proliferation, so expansion, and then differentiation. We have to get some plasma cells, which are the effectors of humoral immunity. And so that's what's happening during that lag period. The first antibody that's going to be made by those plasma cells is IgM, and they're showing you that with the red line. We're going to get class switching, and we're going to get some IgG. And so then we see the IgG titer go up. But what's happening in this lag period, that is when a person, all they have is their innate immune system, and that's usually not enough to give immunity, so that person's going to have signs and symptoms and they're going to get sick. And then here, now we were talking about the secondary immune response. So the secondary immune response is a response of the adaptive immune system to an antigen previously encountered with. So second time, third time, fourth time, fifth time, that person's been exposed to it over the course of their lifetime, we really don't call it a different thing. It's just secondary immune response. And so it's going to be mediated by memory cells. And the big thing, so look down here, so now we're seeing the second time an antigen is produced. Look, so very, very short. There's still always going to be some remaining antibodies is what they're showing you. So there's some slight antibodies probably. But then you see this very fast, very rapid antibody immune response. And so that should be enough to provide immunity because those antibodies are going to bind to something, smother it, neutralize it, and target it for destruction. Pop down here because also think in mind that a person has a billion B cells or T cells. We're just looking at B cells right now, but just kind of like it transfers to T cells as well. And so there's always going to be some of those B cells that didn't get encountered last time, that didn't get activated. And so that's just what you're seeing with this IgM response. And so in case you're wondering, like, well, how come there's another primary response? And that very well could be the case. So what a vaccine does is when someone is vaccinated, that is a primary response generated to something that would do no harm, that wouldn't make someone sick. And so we'll talk about vaccines in the next unit. And so that primes someone's adaptive immune system. So the idea is that through vaccination, when the person really comes in contact with that antigen, they're going to already have a fast, rapid response that's going to give them immunity. And so that's, like I said, where we're headed next unit. So when you get a vaccine, it's artificially acquired active immunity. Just planting a seed for later. Okay, it's just sort of a picture of what we just said. It shows that the antigen is meeting the B cell. It also has to get those cytokines, so that activation from the T cells, the T helper cells. So then we go through proliferation and differentiation, and it's going to be the plasma cells that are making the antibodies, and the memory cells are going to stay behind for that secondary response. Keep in mind, those proliferating cells, they're still going to go through affinity maturation and class switching. But like I said, the big thing with secondary immune response, future exposures elicit an even stronger response. So it's just going to continue to get greater the more times a person is exposed to the same antigen. Look, there's a picture that basically says the exact same thing once again. So if you like this picture better, I'm going to let you go ahead and look at it. I think I've kind of said it enough. All right. So that was for T-dependent antigens, and that's by far the majority of them. Those are going to be protein antigens. B cells, some of them can be activated without a T helper cell, these, like I said, relatively few antigens, and I'm just going to skip right ahead to what's medically important about it. LPS is the gram-negative cell wall, and that is an example of a T-independent antigen. The thing is, small children, young children, they are not capable of mounting this T-independent response. And so it's just another one of those reasons why children are in that immunocompromised group. They haven't built up their adaptive immunity, and then two, it's just they're not capable of that T-independent antigen response. All right, so hopefully you feel pretty comfortable, or at least are beginning to understand that humoral adaptive immunity. So now we're going to flip to the other side. So T cell response and that cell-mediated immunity. So first we're going to look at the receptors on the surface of T cells, and then we'll look at the receptors on the surface of antigen-presenting cells and just host cells. So first let's look at the T cell. And so this is just sort of a really simple version of the T cell receptor. And so a T cell receptor, remember, T cells can only respond to antigens that are displayed within a barcode. So on a host cell or on an antigen-presenting cell. And so they cannot respond to just free antigens laying around, so free virus particles, bacteria, or toxins. And so if you look over here, this is, I guess, the more complex version. Notice that a T cell receptor is going to be made up of an alpha chain and a beta chain, and it's going to have a variable region, much like the hands of an antibody. And so just instead of, antibodies are Y-shaped, they have two hands. A T cell receptor only has one, but they have multiple on their surface of the T cells. So the variable region, and then that constant region is going to keep it embedded into the cell membrane. So big thing about T cells, antigen must be presented by another cell. They are that antigen has to be held in the major histocompatibility complex molecule on the surface. There's two different flavors of MHC molecules, which we're going to look at in the next slide. But antigen-presenting cells, so dendritic cells, macrophages, B cells, they have an MHC class 2. They are going to be displaying exogenous antigens, so things that they've picked up in the tissues, in blood, in secondary lymphatic tissue, things they've eaten. Remember, they eat things, they digest them, and then they display a little piece on their surface, much like cutting the head off of a vanquished foe, and then they bring it to the adaptive immune system, to those T cells. Since cytotoxic T cells, their job is to respond or to eliminate basically invaders inside of a host cell. So either a virus inside of a host cell or a cell that's become tumorigenic. And so corrupt cells. And that is going to be through what's called an MHC class 2 molecule. So viral infected cells, tumor cells. So these are endogenous antigens. And so the cytotoxic T cells are going to respond to endogenous antigens, not the stuff laying around. That's the job of the antibodies. And so this is just sort of a summation, just to remind you about the CD8 cells and the CD4 cells, so cytotoxic cells and helper cells. This up here is looking at a major histocompatibility complex. And if you take that word apart, histology was the study of tissues, compatible complex. And so these are our barcodes. Yours looks different from mine. The only people on the planet that have the same major histocompatibility complex are going to be identical twins, because you get this genetically. And just due to, once again, genetic reassortment of genes, there's multiple ones, everyone has their own unique major histocompatibility complex. So people in families tend to have the same one. So these are the things that need to be matched if someone's going to donate organs or bone marrow. And so another name for them is human leukocyte antigens. So it determines tissue compatibility, so self-antigens. So looking over here, all of the nucleated cells, including antigen-presenting cells, so everything but basically your red blood cells, have an MHC class 1. Okay, this would display something if this host cell were infected, so MHC class 1. On antigen-presenting cells, they have MHC class 2 molecules also, because they also have a nucleus, so they still have MHC1. So this is their antigen-presenting barcode. So the two messages that can be sent, so pop down here for a second. So cytotoxic T cells, their antigen recognition peptides presented on MHC class 1 molecules. Their job is to kill cells that have been virally infected or tumorigenic. So when they meet their antigen that they're designed to respond to in the context of this MHC class 1 right here, they come alongside a cell and induce that target cell to undergo apoptosis. And so their source of antigen is endogenous, produced within the target cell. So when a virus is infected in a cell, that cell will put out a little piece of the virus. If it's a tumorigenic cell, it too will have tumorigenic markers. The helper T cell is a little different. Remember, that's the one that's the helper for both B cells and T cells to become activated. It is going to be talking to the antigen presenting cells. So it reads its antigen, or it gets activated by its antigen in the context of this MHC class 2 molecules. And so when an antigen presenting cell brings an antigen in this little groove right here, presents it to a helper T cell, it's like saying, hey, look what I found in the tissues. It's foreign. Help me mount an immune response against it. And so that's sort of the message the antigen presenting cells send when they display the antigen in an MHC class 2 receptor. Okay, and this is just kind of showing you what I said. So don't forget, so CD8 cells are the killers. So they always have that CD8 marker in conjunction. It's next to their T cell receptor. So they're the ones that respond to a sick cell. So a cell virally infected or tumorigenic. And so it's just showing that the MHC1 with that antigen on its surface. A CD4 cell, remember, is a T helper cell. So it has that cell determinant 4 marker alongside its T cell receptor. It responds to antigens being presented with that presenter barcode, antigen presenting cell, so an MHC class 2. Just kind of want to draw your attention to that CD4 was one of the receptors for the virus that causes AIDS, the human immunodeficiency virus. So that's how it enters and infects a T helper cell, which is that pivotal cell in the immune system. All right, so in case all that was just jibber jabber, here you go.
Macrophages and dendritic cells are major antigen-presenting cells. After digesting an antigen, they process parts of the antigen, attach them to MHC2 proteins. These are different from the MHC1 proteins which most body cells possess. MHC2 proteins identify the macrophage or dendritic cell as an immune cell, so that if it presents an antigen, it won't be destroyed. These MHC proteins then migrate to the cell surface and thus wear the antigen on the surface of the cell. This presenting of the antigen allows antigen-presenting cells to activate the T lymphocytes in the adaptive immune response. Some dendritic cells are able to enter lymphatic vessels after processing an antigen and then exit in lymph nodes where they can present to T cells there. So it is this return from peripheral tissues to lymphatic centers which allows these dendritic cells to play a special role in the maturation of T cells based on experience of antigens in the peripheral tissues.
Okay, so that just kind of gave you a reminder of those specialized antigen-presenting cells, the dendritic cells in the tissues. They collect their antigen, they eat it, and then they're going to display it, and then they go to those secondary lymphoid tissues and then present them to the T cells. And so just like we just saw, we have those presenting cells. They have a co-stimulatory molecule that can activate those cytotoxic T cells. Still, the cytotoxic T cells still need cytokines from the T helper cells. But the idea is antigen presenting cells going to present them to these naive T cells that recognize that antigen, and then they talk to each other through those cytokines. But as long as you've got antigen presentation and that activation through co-stimulatory molecules, then we get activated T cell proliferation and differentiation. Over here, once again, it's showing those dendritic cells. If they are presenting things that are self antigens or things that are harmless, there won't be any co-stimulatory molecules. And just like when we don't get that activation with the B cells, same thing with the T cells, what's going to happen, you can get anergic T cells. And so remember, without energy, these are unresponsive cells. And so they too, instead, they're just going to undergo apoptosis, just like those anergic B cells. And there's also regulatory T cells that are involved, but just prevent certain immune responses. So just kind of reminding you, in order to get activation, in order to get that proliferation and differentiation, you have to have both the antigen and activation through that co-stimulatory molecules, which are cytokines. Okay, so this is the text version of what I just said, but I kind of want to expand and kind of remind you, going back to innate immune system. So remember, the way that these innate immune cells recognize that something is foreign is through those toll-like receptors. And so they have some idea of what they've eaten. So whether it's that antigen attached to the toll-like receptor that detected LPS, or whether it detected flagellin, and then there were others that responded to viral RNAs. So there's going to be that signaling. So when they talk to each other, that antigen presenting cell is giving information to. So whether it's a dendritic cell or macrophage, they're going to release cytokines going, hey, by the way, this antigen, I know it's bacterial, or hey, this is a helminth, or hey, this is a virus particle. And so that's what's happening through that communication of those cytokines. And so that's going to help mount a specific T cell response. But like I said, you always need that co-stimulatory molecules that signals that whatever that antigen was eaten is dangerous, and then we're going to get that response. If you don't get that co-stimulatory signal, so the T cell is going to become anergic, goes through apoptosis, or it can develop into a T regulatory cell. And so once again, they don't really know a whole lot about these regulatory cells. They just know that they're there and they're involved in preventing autoimmune diseases. Okay, so yes, I've made another big messy diagram. So just kind of building upon it. So activated T lymphocyte populations, exposure, activation, the effectors, we got our cytotoxic T cells, our helper T cells, we get regulatory T cells. I want to look right here in this helper T cells. So once again, the helper T cells, those are the communicators, those are the talkers. And they're going to release cytokines talking to macrophages, B cells, and other T cells. And so there's different populations of these T helper cells. And they know, based on whatever the antigen presenting cell said through cytokines, because of whatever toll-like receptors were stimulated. Remember, those toll-like receptors respond to PAMPs and DAMPs, so pathogen-associated molecular patterns or damage-associated molecular patterns. So they have information, so they kind of know what they're presenting. And so they talk to the helper T cell, and that causes that helper T cell to become either a Th1, a Th2, or Th17. And so they know that Th1 cells, they're really great against intracellular invaders, so like viruses. Th2 cells, they know that those T helper cells, they're really effective orchestrating a response against parasitic worms. And Th17 cells, they know that they're really effective against extracellular invaders, so like bacteria. And so I'm just saying this is just one of the ways that the innate immune system communicates with the adaptive immune system and make sure that the right immune response gets activated. So this is one of my favorite videos. I know I have a lot of them, but this is actually some beautiful fluorescent microscopy work. And it's actually going to look at the method that cytotoxic T cells and, spoiler alert, natural killer cells use to kill your self cells that are corrupted, so viral infected cells or tumor cells.
Inside all of us lurks a serial killer. A killer whose primary function is to kill and then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They patrol our bodies, identifying and destroying virally infected and cancer cells, and they do so with remarkable precision and efficiency. There are about 5 million T cells in a teaspoon of our blood, engaged in the ferocious and unrelenting battle to keep us healthy. These amorphous blobs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds a cancer cell, membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs of cancer. They kill their targets using poisonous proteins visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins. This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
Okay, so how do they kill? So it's just kind of showing you how CD8 cells, those cytotoxic T cells, kill. So they're the ones, once they're activated, they're going to go along and they are looking for, this is showing you a virus-infected cell, and they are displaying a viral antigen in the context of their major histocompatibility 1 cell marker, so self-antigen. And so it's going to come alongside, and just like you saw, first there's perforin that's going to poke holes. Think about the edges of your paper are perforated, and then it releases, your book called them proteases. If you took AP2, that book called them granzymes. Either way, what these proteases do, they go inside of a cell, they're going to start breaking down proteins, but basically they're going to induce apoptosis, so that programmed cell death. And so it's nice, it's neat, it's orderly. Remember, they're just going to sort of basically chop everything up and bleb up into little tiny packets, meaning not release that intracellular pathogen. So don't let loose of those virus particles, don't let loose of that bacteria, whatever it may be, intracellular. And then the macrophages will come along and eat it. A activated cytotoxic cell, it can then also release cytokines to alert the neighboring cells. And so just once again, the cells of the immune system, they communicate through cytokines. All right, so what do the effector CD4 cells do? Once again, activate, direct, and support. So once they are activated, they release cytokines. And so they respond, they have to be presented, so that antigen presented in context of that major histocompatibility class 2. They're going to activate B cells. B cells then will become plasma cells, produce antibodies, activate macrophages. How do you activate a macrophage? Oh, it gets bigger. Increases its metabolism, number of lysosomes, that's how it dissolves things, nitric oxide, other toxic compounds, that's what it uses to kill. 2, macrophages can form a really big cell, so that's what happens in granuloma formation when they can't kill it, but they can wall off that part. We saw that with tuberculosis, just to protect other cells, and then they also release cytokines that stimulate cytotoxic T cells. This is kind of showing you, so this is the T helper cell. It has to be presented with that antigen, but then it's going to make cytokines that, it stimulates itself too. A little self-stimulation there. But then it is also responsible for stimulating cytotoxic T cells, so you get that cell-mediated immunity attack on infected cells, and then also those B cells, you get that humoral response with antibodies. And so this is the cell in that that is infected by HIV. When you lose the T helper cells, when that goes down, that's when someone has AIDS, acquired immunodeficiency syndrome, and that's when they lose their immune system and they end up dying of an opportunistic infection. All right, so we're wrapping it up, so hang with me. This natural killer cell, they are actually part of the innate immune system. So they are not specific, meaning they don't recognize a particular antigen. They do kill viral infected cells or tumor cells by cell-to-cell contact in the same way using perforin and those proteases, granzyme. They have two distinctive protective roles. They can come alongside self cells. So this is a body cell that has antibodies attached to it. And so then that just tells that natural killer cell this is a bad cell. So it's called antibody-dependent cellular cytotoxicity. So once again, antibodies bind to things and target them for destruction. And so those natural killer cells, they have Fc receptors, so they can bind to these that constant region, or the stem, if you will, of the antibodies, and then they're going to kill by cell-to-cell contact. Or stressed cells that lack a MHC1. And so what happens, the reason why we really need these natural killer cells, some viruses, like herpes virus and some tumor cells, they can actually downregulate MHC class 1. So they make those host cells not have as many MHC class 1. And remember, cytotoxic T cells have to see the antigen in the context of that MHC class 1. And so that downregulation allows them to escape from cytotoxic T cells. And so it's kind of cool that the natural killer cells, they don't need to see that antigen. They just see, hey, you don't have an MHC class 1, or that MHC class 1 isn't right. And then they can kill using perforin granzyme once again. So it's just kind of showing you viral genome keeps that host cell from displaying that MHC class 1. Without these cells cannot be a target for the cytotoxic cells, but the natural killer cells can then come along once again and induce apoptosis. So just kind of keep in mind, natural killer cells kill like T cells, but they are non-specific. They are part of your innate immune system. All right, I think this is the end, but way back when, it seems so long ago, we talked about how some of the damage done by disease is through collateral damage or through damage from the immune system. And so now that we've talked about it, it's going to make more sense. So damaging effects by inflammation. Remember, we have phagocytic cells releasing enzymes, toxic products, and we know that inflammation causes a lot of the things associated with bacterial meningitis, and certain sexually transmitted infections cause a lot of that damage. Because with inflammation, we looked at it, chronic inflammation is bad and causes damage to surrounding tissues. Damage associated with exaggerated or inappropriate immune responses. The cytokine storm we talked about, so that overproduction of cytokines, exaggerated immune response. So you get fever, body aches, can also lead to shock. And so that's what happened in the 1918 flu pandemic, and that's what's happening with the 2020 COVID-19 pandemic. People dying from that cytokine storm. You can also get immune complexes. So now we kind of know what that means. Those antigen-antibody complexes can form, settle in kidneys and joints, and that's sort of the cause of that acute glomerulonephritis following skin throat infections with Streptococcus pyogenes once again. So then we end up with kidney damage. Autoimmune response cross-reactive antibodies, once again, Streptococcus pyogenes is huge for that, just because they have that capsule of hyaluronidase, which looks like our tissues. And so that's what's causing that acute rheumatic fever, and that autoimmune response is attacking the heart and also attacking joints. So hopefully those things make a little more sense now. I told you I was going to bring them back up when we actually studied the immune system. And then that last part, super antigens. And so this kind of ties back with T cells, and I think it'll make a whole lot more sense now. So this is our last topic.
Antigens are normally processed by antigen-presenting cells and presented to T helper cells on class II MHCs. Only the T helper cells with T cell receptors capable of interacting with the antigen are stimulated to produce cytokines. The activated T helper cell stimulates only B cells that react with the same antigen, thereby limiting the response to the production of antibodies that were presented. Superantigens such as the toxic shock syndrome toxin produced by Staphylococcus aureus are not processed by antigen-presenting cells. Instead, they bind directly to the outer portion of an MHC class II antigen on antigen-presenting cells and to the outer portion of the T cell receptor of T helper cells. Rather than binding to only the cells specific for that antigen, which represent about 1 in 10,000 T cells, the superantigen binds without antigen specificity to as many as 2% to 20% of the T cells. This results in stimulation of many T helper cells. The result is the release and activity of excessive amounts of interleukin-2, IL-2, and other cytokines that enter the bloodstream instead of only acting locally as they normally do. This leads to symptoms of fever, nausea, vomiting, diarrhea, and sometimes shock, with failure of many organ systems, circulation failure, and often death.
Okay, so the superantigens cause that cytokine storm because they can get those T cells excited in a non-specific manner. Okay, so that was it. That wraps up this unit. I hope you have an awesome day. As always, if you have questions, I'm here for you.